Okay, so if you're looking at those questions on the review sheet I just gave you, those are the questions that I gave you at midterm. So you may already have those together. I don't know. Okay, so we said the 4 steps necessary for life. We already said the first one, make monomers, like amino acids and nucleotides. Okay, what's the second one? And this is on your first page of notes. Good. Yep. Monomers together. You got to put all those monomers together into polymers. Okay, so monomers put together into polymers. Example of a polymer. Come on.
Proteins.
What'd you say? Proteins? Good. So we're going to take amino acids made in step 1 and put them together and make a protein. Does that make sense? We're going to take nucleotides and put them together and make what? DNA or RNA. Good. Okay. Why would we make RNA first? Why do they think that RNA evolved before DNA?
It's simpler, it's not helpful.
Absolutely. RNA is single-stranded. It's unstable. And so we took the RNA and then eventually made the DNA. So evolution was backwards to how we do it now. Okay. Back to the step 1 where we made the monomers. How did they do that? How did they make monomers from nothing? Or how do they think? You know, this is just a theory. So how do we think? Yep, look at this right here. Right here. Miller-Urey experiment. Do y'all remember this? Okay, so you took right here, you put like electric shocks in it, which was like the lightning, so an energy source. What did they start with? Like, what did they do to start? Bingo, absolutely. So somebody said they took inorganic molecules, which are non-living things, and they did not make living things, but they made the little pieces of living things. Like, so they made nucleotides or amino acids. But Annika nailed it. The inorganic things, they took what they thought early Earth was. So early Earth. So they took like gases that they thought were on the Earth, like methane and nitrogen gas and water, and put it together with lightning and steam, and they made amino acids from that. Okay, one thing you got to know, what gas was not there? Oxygen. Boom, good. Oxygen was not there. Okay, good. Okay, so step 1, they made monomers with the, what's this experiment called? Miller-Urey experiment. It's almost always in the multiple choice somewhere, so kind of get that. It seems like y'all got it. Okay. Step 3. What was that? What would step 3 be? Yeah. Okay, so we already made the DNA and RNA, but step 3 says we have to be able to replicate it, right? And I'm just going to put RNA replication so that you can remember that RNA was first. We got to be able to copy DNA and RNA. And then 4, we got to make a cell. So if you're looking at your handout, that second word, protobuoyant, that's where a cell membrane formed around the cell. I tried to choose words that I didn't think you would know. Yeah, this is yours to keep. Okay. You can write on it, or you can write on another sheet of paper, whatever.
What did you say?
Proto, that second word is when a cell membrane formed. So y'all see, it's a lot of work, but I'm going to help you as we go. Okay, step 2, or question number 2. What is the importance of surface area to volume ratio and why? Go. Anybody?
You want more surface area so that you can have volume. You want less of because
Absolutely. You want more. Oh, that was not right. You definitely want more surface area. Bigger surface area so that you can have, she said it, more space to get food molecules in and waste molecules out. Food molecules in, waste molecules out. And my example for this is always the school. We have one entrance, really, I guess two entrances for 1,600 people to leave at 3:15. That's not smart. That's why traffic is horrible at 3:15. If you wait till 3:20, You might as well wait till 3:45. Because y'all know you're not going anywhere. Yes, ma'am.
More necessary to get food molecules.
Yeah, to get food molecules in and waste molecules out. So cells need a higher surface area. Cells need a higher surface area. Okay, what are some things in your body that have a high surface area? Intestines. Your intestines are all crooked, right? So you can get more food that way. Like they're all folded. Like there's more outside than there is inside. And also they have cilia inside them. That little hair that allows it to trap the food so you can absorb more food. Okay, what's another thing in your body that has increased surface area? Skin. It's got all the folds, like your fingers and all the folds, but we don't really absorb anything through it. Yeah, like hair, that would all be, what'd you say? ER, your endoplasmic reticulum is all folded up. Lungs, right? Lungs have the alveoli in them, so you can get more what? Oxygen. So the higher the surface area, you can absorb more, or like take in more particles. What's something not in our body, like maybe in a plant that has a high surface area? What'd you say? Well, yeah, the thylakoid would have a high surface area. She said chloroplast. Roots. Remember I told you about the roots? What's wrapped around the roots to help them absorb more water? What's wrapped around the roots of a plant to help them absorb more water? Fungi. Mycorrhizae. Fungi. So around all the roots, there's a fungus wrapped around them. And that fungi helps them to absorb water. Y'all remember that? And that's called mycorrhizae. Okay, so moving on. You know how to calculate it. Look at the formula sheet. I think you all have that. Okay. Explain the difference between potential and kinetic energy as it relates to bonds. Yeah, absolutely. Potential energy is energy that is stored in bonds. Like, what's an example of something stored, energy stored in?
Battery.
What'd you say?
A battery is not plugged in.
Yeah, a battery, but something you eat. Like a carbohydrate or fat, right? So in that carbohydrate, like when you eat a potato, in those carbohydrates, the bonds are storing the electrons, and the electrons are energy. You know, think about electrons being energy. Electron transport chain is pumping up those electrons and hydrogens, getting energy. So the bonds of the carb is where the energy is stored. When you break the carb, you do what? When you break the carb, you release energy. Very good. Absolutely perfect. Okay, so let's look at this for a second. Okay, the difference in... what's it called when you break a carb? Catabolism. I'm horrible at writing on this thing. Okay, what's it called when you build a carb? You don't build carbs. When you build DNA. Good. Y'all are so smart. Okay? So when you... Catabolism, we said, is breaking bonds. And it does what with energy? Releases energy. Did I break somebody's pencil in here? Whose pencil did I break? Oh, I broke my own pencil. So think about a pencil as like a carbohydrate. When you break the pencil, sound energy was released. When you eat a piece of potato, you break the carbohydrates, the energy is released from the bonds. Okay? And then when the energy is released, it's moving around. That's kinetic energy. That's the answer to the question. Okay? Anabolism is where you build molecules. What do you build in your body? What do you have to build? Two things. DNA and protein. Good. So I had pizza last night for supper. I broke that down, and now with those materials, I'm building new proteins and new DNA. Okay? What does this do? Anabolism, what does that do with energy? Stores energy. Awesome. Okay? There was another word I told you that meant catabolism. What was that word? It's the same thing as, what'd you say? Y'all, Lex is all over this. She got it. Hydrolysis. And it requires water, right? It breaks lysis with water. What about another word for anabolism? Man, y'all are ready. Dehydration synthesis.
says like dehydration/condensation.
Yeah, it's the same thing. That was what's in the notes. Dehydration or condensation reaction. Okay, so y'all ready for the next question? Name the theory that supports the evolution of prokaryotes to eukaryotes. Name 3 facts that support it. What's that theory? Endosymbiosis. See this picture right here? Okay, always on the test somewhere. Endosymbiosis. Somebody tell it to me. Yep, it's when a big bacteria engulfed a small bacteria. It's a theory. A big bacteria engulfed a small bacteria. And what did that little bacteria eventually evolve to become? The mitochondria or the chloroplast. And what are some things that prove that? What are some facts for that? Yeah, so, okay, a mitochondria, so imagine that there's a mitochondria right down here. So, mitochondria and a bacteria both have their own DNA, they both have their own ribosomes, and they both do what? Mitochondria and bacteria both, what'd you say? No, glycolysis is in the cytoplasm of everything. They what? Yeah, they copy by themselves. What's it called when bacteria and mitochondria copy by themselves? What? Y'all are supposed to know this. Bacteria, when they... Binary fission. Okay, so bacteria divide by binary fission. Mitochondria divide by binary fission. Yes? Okay, so... Okay, look at my picture up here. This little bacteria right here and this mitochondria, they're like, what do they have in common? Well, they both have their own DNA. So mitochondria have their own DNA separate from the DNA in the nucleus. It's like DNA for making ATP. Mitochondria and bacteria both have their own ribosomes. They both divide by binary fission. And it's not just the mitochondria, it's what other organelle? Chloroplast. Good. Okay, so y'all got endosymbiosis? I feel like y'all do. We've talked about that a lot. Okay, the next one. Name 5 organelles found in eukaryotic cells. State their function. The function of these organelles are necessary in prokaryotes. How do prokaryotes perform these functions without organelles? Okay, instead of answering this question, because it's long, I'm going to draw this, and if you want to draw it, you can do that instead of answering it. What is it?
What was the set for the three evidence?
Well, the evidence is it was like they both have DNA, they both have ribosomes, and they both do binary fission. And you can actually we just said 6 because the mitochondria has DNA, ribosomes, and binary fission, and so does the chloroplast. That's really 6 evidence we just said. Okay, so draw you a big old circle like on another sheet of paper. And I have good news. This is the last time you'll have to see me do this. Okay, so the big circle is the cell membrane made of what? Okay, so the structure, you need to put this on your paper. The structure of the cell membrane is phospholipids. Yeah. The structure of the cell membrane is phospholipids. What's the function of the cell membrane? It lets things in and out, right? What does it let in?
Small uncharged particles.
That's right. Small uncharged particles. Good job. So the cell membrane, I'm taking all this off. I'm starting over. Is made of phospholipids and proteins. So that's the structure. The function is that it lets small uncharged particles in. It really does all transport. But if it's a large particle, how does it get in? Through a protein channel. Boom. Good. Okay. The next one we're going to talk about is the nucleus. What is the structure of the nucleus? Yeah, it's basically the cell membrane, but around the nucleus. It's a nuclear membrane. It's got nuclear pores in it. What needs to come out of the nucleus? Why would it need to have pores? The RNA. Good job. See, this is going to be easier now that you know it. Okay, what's inside the nucleolus? Genetic information, right? So the function is to store the genetic information. In the form of, what's that in the form of? No, you got it. A chromatin. Chromatin, which is DNA loose, is that acetylated or methylated? Acetylated. Good. Okay, a chromosome, if it had a chromosome, that would be good for what? Methylated. Okay, so just a little, don't draw this, but just a little by the way. Chromosome is loosely coiled, it's acetylated. Why is that beneficial? What would chromatin? Yep, that's your chromatin. Okay, it's loose so that you can access genes. So you can do DNA replication or you can do transcription when it's in chromatin form. Okay, a chromosome is when it's like really tightly coiled in the shape of an X. What's good about that? Division, mitosis. Not DNA replication, because if you were trying to get, look, this makes perfect sense. If you were trying to get that one gene right there, that little black dot, that one right there, you can't access it when it's all chromosomed up and coiled tightly. But when it's like this, you're like, oh, it's right there. Loosen, acetylated. So this is methylated, that's acetylated. Okay, so now back to my cell. Okay, so we know that. Let's go with, what does the nucleolus do? Yep, nucleolus is where, what's inside the nucleolus? RNA, good. So it's made up of RNA, and it is RNA. It is RNA. The nucleolus is RNA. It's a dense ball of RNA. It's where ribosomes are made. What are ribosomes made of? RNA. That was not a trick question. If the nucleolus is made of RNA and ribosomes are made there, then the ribosomes are also made of RNA. Okay, so next, let's go with right outside the nucleus, the rough ER. Okay, if I get to write about the structure of the rough ER, it's made of cisternae, which is basically cell membrane flattened out into tubes. But if I get to write about that, I'm saying it's got ribosomes attached to it. The structure is it's got ribosomes attached. Okay, what is the structure of the rough ER? We're going to say ribosomes attached. What's the function?
Package and send out materials.
What is it?
Package and send out materials.
Not really package, but send, transport. So the rough ER transports, what does it transport? Proteins. Transport proteins. Move the proteins around. Okay, the smooth ER, which is the same thing but right outside it. What's the difference in the smooth and the rough?
One transports proteins out of the cell. One.
Smooth ER doesn't have the ribosomes attached. Look at my picture. It's like, oh, that's easy. Smooth ER doesn't have it attached. Okay, the smooth ER, I feel like, does a whole lot of work and never gets any credit. Nobody remembers the smooth ER as an important organelle, do you?
Yes.
You do?
Lipids.
Okay, it has a lot of functions. Y'all tell me the functions. Let's go.
Detox.
Detoxes blood.
Sports livers.
Stores calcium. Breaks carbs, and it makes phospholipids. So y'all take a second and try to remember that. Detoxes blood. Stores calcium. Makes phospholipids. Breaks sugars. Breaks carbs. And that's all the smooth ER does. So, okay. What type of cell in your body would have a lot of smooth ER? Liver cells. Good. Because liver cells detox blood. What'd you say? Maybe?
You have to say your intestines takes toxics out of your
What was
face.
this last one, Ms. Gilbert? Detox blood, stores calcium,
Breaks sugar. Did you get that one? Makes phospholipids.
and break sugar.
Okay. The next one, very important one. The mitochondria. What would you say about it? It's made up of cristae. Okay, cristae, cisternae. Those are two different words. Get them separated. Cisternae, CI, is what makes up the rough ER and the smooth ER and the Golgi. Cristae, which is an important word, which may be on the test. Cristae. Cristae. That's the mustard on the hot dog of the mitochondria. Hey, it looks like a hot dog. Cristae? Okay, somebody tell me what is... So we got the structure, but you could have said double membrane, on ribosomes, cristae, shaped like a hot dog. No, don't put that. Okay, what's the function of the mitochondria? Thank goodness we know that. I would have been real sad if we didn't. Make ATP, right? Make ATP. Somebody tell me the 3 steps to making ATP. Glycolysis. Boom. Glycolysis, Krebs cycle, etc. Look right here. Glycolysis happens outside. Glycolysis. Krebs cycle inside. etc. happens where? On the cristae, in the electron transport chain. The cristae is the electron transport chain. It's like cell membrane folded up in the mitochondria. High surface area, lots of ATP. Good. Okay, let's say this is not an animal cell that it looks like. It's a plant cell. Or not a plant cell, it's a algae. So it's got a mitochondria and a chloroplast. What's the structure of the chloroplast? Good. You remember the thylakoids? What's the thylakoids are like cookies, right? And the whole stack is called grana. And outside the thylakoids is called stroma. Okay. What is the function of the chloroplast? Photosynthesis. What else? What would you say? The function is to make sugar or photosynthesis, either way. Okay. What about a lysosome? It's got digestive enzymes to clean it, and it cleans the cell with lysozymes inside it. What kind of cells would have, in your body, what kind of cells would have lots of lysosomes? What'd you say? White blood cells. Like macrophages? Macrophages are the white blood cells that eat bacteria and chew them up. Structure of the chloroplast has thylakoids. Okay, what if this cell had cilia on it? For movement, right? Or I feel like the test always says locomotion, which means movement. Okay, so let's go with still answering this question. You can't do this to your picture. Oh no. It's gone. Okay, so a bacteria doesn't have a nucleus, but it does have DNA, right? So there's no nucleus, so the DNA is just in the cytoplasm. There's no mitochondria, so how do they get energy? No, you don't have to. You don't have to draw this. I was just trying to help you understand. So they have DNA, but no nucleus. They have mitochondria, they do not have a mitochondria, but they can make energy. How? Enzymes. Do they do glycolysis? Yes, bacteria do glycolysis right here in their cytoplasm. And then where do they do the electron transport chain? Boom, right there in the cell membrane. Do y'all remember that? Yes, everybody remember that? And the bacteria do the electron transport chain in their cell membrane. That's cool. They do glycolysis right there in their cytoplasm. They do electron transport chain in their cell membrane. Okay, what about, do bacteria have ribosomes? Yes. Why do they have ribosomes? To make proteins. Oh, we didn't talk about the structure of a ribosome. What is that? What are ribosomes made of? 2 subunits, large subunit, small subunit. They're made up of rRNA. And what goes through them? mRNA goes through them, right? And what brings the amino acid to it? Y'all are so smart. tRNA? It's too bad it won't be my drawings on the AP test. Then you'd for sure make a 3. Okay, so in your textbook is a table that's like this, and it's all the organelles, all their structures, and all their functions. And there's more. And that's the picture out of the textbook. So if you want to look at that tonight, do so. Okay, next question. So back to the original questions. I think we got them all. Any questions on those questions? Any questions on those? Okay, next set of questions that you're looking at. What is the structure of the plasma membrane? What is the function of each individual component? Okay, there's your plasma membrane. Somebody tell me what is the, there's the structure, it's showing it. But what are some of the parts? Okay, phospholipids. Yeah, what are those little indicators called? Glycoproteins. I'm going to put carbs because remember that a glycoprotein is a carbohydrate attached to a protein. Or a carbohydrate attached to a lipid is a glycolipid. So, okay, just a little by the way. Glycoprotein right there. Sugar attached to a protein. Glycolipid right there. So glycoprotein, glycolipid. Okay, so we got carbs. What are they for? Recognition, right? Hey. Like cell membranes recognize because of their carbohydrates. Okay, what else? Phospholipids, carbs, cholesterol. What else? Proteins, like the big ones. Okay, so let's start at the top. Just a phospholipid. Tell me everything you know. Go.
Polar head, non-polar tail.
Polar head.
Non-polar tail.
Non-polar tail.
Unsaturated, no saturated.
All right. Okay, so so the top is polar, and we'll draw a pole through it. Hydrophilic, so it likes to drink water, but the feet are hydrophobic. Okay, so just to make sure you got this. The head, hydrophilic and polar. The tails, hydrophobic, non-polar. What does the word polar mean, Lex? Somebody help her out. Has 2 charges. The word polar means, okay, that's one of your vocabulary, so write it down. The polar means it has 2 charges. It's got a positive side and a negative side. Example would be water. The hydrogens are positive. The oxygens are negative. So water has 2 charges. It's like half positive, half negative. That's what polar means. Okay. So the head of the phospholipid is what? Polar and hydrophilic. Y'all remember that? Hydrophilic. What does that mean? Likes water. The tails are hydrophobic. Okay. The only reason that's important is because it lets some things in and some things out. Like some molecules can come into the cell because of this polar, non-polar water, not like water layer, and some things can't. So we said small uncharged particles can come in. Large charged particles, aka ions, can't. If an ion's going to come in, how's it going to come in? A protein channel. What'd you say? Ion channel, protein channel. Good. Okay, so we got phospholipids. The carbs, we already said they're for what? Recognition. Carbs attached to proteins and lipids, glycoprotein, glycolipid, and they're for recognition. Okay? Cholesterol, what's that for? It's to make it not hard, jelly-like, to keep it from freezing, to keep it, I think the word in your notes is fluidity. Like to keep it fluid, right? Y'all remember that? All right, the proteins have a lot of functions. What are they for? So these purple things, the purple things in that picture, They're for recognition, for cells to stick together. What else? Anchorage, cells sticking together, recognize each other. What type of protein helps recognize? What type of protein helps? Glycoproteins help recognize, right? With their sugars on top. Okay, so glycoproteins help recognize. They stick together. I think the most important one is what? Transport. Like the ion channel, letting things in and out. Y'all remember that? Letting things in and out. Like, okay, y'all remember when we did the neuron and we sat on the floor and we did the sodium-potassium pump? That was a protein pumping in sodium and potassium. And then we did the gated ion channel. Okay, what about on test 3 when there was a protein that receives a message? What was the name of that protein? G protein, right? Y'all remember the G protein accepted the ligand? That was a protein in the cell membrane that received a message. So you got that. Okay, next question. What are 4 macromolecules and the composition of each? Okay, so 4 major macromolecules are what? Proteins, nucleic acids, lipids or fats, and carbs. Good. Okay, so let's look at these pictures over on the side. There's going to be words on this slide. So these are 4 biological, like 4 big macromolecules that are important. Okay, the first one, look at this picture right here. That's a phosphate, a sugar, and a nitrogen base. What is that? That is a nucleotide, which makes up nucleic acid, right? So nucleic acid, nucleotide. Phosphate, sugar, base. If you don't know that, after I made you color them, cut them out, and tape them together, I need to quit. Y'all remember taking these and coloring them and taping them together and then making a whole class of DNA? So DNA is made up of nucleotides, phosphate, sugar, base. Okay, anytime on the test you see the word nucleic acid, I want you to think that means DNA and RNA. That's like just a general term for nucleic acids. So DNA and RNA are made up of, like, what's their monomer? The monomer is a nucleotide. The monomer is a nucleotide. What's monomer mean? Simple, like one of them. So one little piece is a nucleotide, and all together it makes DNA or RNA. Okay, look at this next picture. This should ring a bell. You've got an amine group on that side. Okay, so here's your amine group. You've got a carboxyl group, a C, O, and an OH. And then you've got the R group. What is that? That's an amino acid. Come on, you got to know that. Amine group, carboxyl group, and then the R group. What is the R group? Yeah, it's something that changes. The R group, there's 20 different ones, and it makes 20 different amino acids. For example, methionine. That's the amino acid that starts making a protein, right? That's the first one. Okay. So it says proteins, aka polypeptide. That's on your vocabulary list. Polypeptide. Proteins made up of amino acid structure and sequence determine function. Where are proteins made? Ribosomes. In the ribosomes. How does your cells know to make a protein? Well, yeah, the DNA is the magic blueprint, right? And the DNA makes RNA, and the RNA is the instructions to make the proteins. So when you were in the 9th grade, and your teacher was, or maybe the 7th grade, and your teacher was like, DNA is the blueprint, and the nucleus is like the brain. And you're like, okay, that's stupid, but you memorized it. Okay, what really it is, unless you have me for 9th grade, because I didn't say that. DNA is really what? It's the code to make proteins. Like, it's got to make RNA first, but then proteins, right? So DNA is the magic blueprint. It's the code. It's the nitrogen base or nucleotide sequence. It's almost like Braille. The ribosome can read it and know what protein to make. And the nucleus is where it's stored. Okay, look at me, because I'm not going to be at your house. You can answer these questions at your house. Primary structure of amino acid is amino acid peptide bond. Amino acid peptide bond. So amino acids are held together with peptide bonds, right? And when it's straight, it's called the primary structure. Okay, I have a picture of it. There we go. So there's your primary structure, that first one. So think about this pot planar being straight. Okay, the secondary structure has alpha coils and beta pleats with what kind of bonds? Please let this be a free response question one day. Please, please. Okay, alpha coils, beta pleats with what kind of bonds? Hydrogen bonds. Okay, now, if I squish this together, tertiary structure, what kind of bonds? Come on. Sulfur. Good. Remember, proteins have sulfide in some bonds. So that's a key. You need sulfide for protein bonds. Okay, when you put more than one of these together, it's called... Yeah, it is the protein. It's a functioning protein. It's the quaternary structure. The structure, folding, is equal to the function. It folds that way because of the way it's shaped. Okay, so seems like you got that. The next one is we already did phospholipid. And then carbohydrate is next. A carbohydrate is a sugar. Carbohydrates are really, really good for you because they store energy. But a lot of them will make you gain weight. If you're not using your stored energy, you'll gain weight with it. Does that make sense? Okay, so we store our carbohydrates in starch. Plants store their carbohydrates in cellulose. Right? I said that wrong. Plants store it in starch and cellulose. We store it in glycogen. I'm sorry. We store it in glycogen in our liver. So like a potato is like starch, right? It's got a lot of carbohydrates in it. You know potatoes or french fries or hash browns, any of that. Got a lot of starch. Okay, next question. What are the factors that affect enzyme productivity? Okay, so what is an enzyme, first of all? A protein with a special job, right? Protein with a special job. Okay, so it's made of amino acids. What affects an enzyme? Like, what would slow down the rate? Temperature? Salinity, pH. Okay, and then there's two types of inhibitors. Say it again. Competitive and non-competitive. Okay, so y'all remember, you got this, you just forgot. Okay, so enzymes, the purple thing is an enzyme. And it acts on a substrate. So amylase in your mouth breaks down carbs. So amylase is the enzyme, the substrate is the carb. Okay? Now look at this picture. The blue molecule. Right here. Blue molecule. That's the competitive inhibitor. Why is it called a competitive inhibitor? Yeah, it's blocking the active site. See, y'all just needed me to remind you. Now you got it. Okay? The yellow molecule is what we call a non-competitive inhibitor. And why is it non-competitive? Boom. It binds somewhere else, the allosteric site. Binds somewhere else and changes the shape of the enzyme. So both inhibitors slow down the productivity. Like amylase doesn't work if it's got if it's got something blocking the active site or if it's got the wrong shape. Amylase isn't going to work. It's going to slow down the rate. Rate. What is rate? It's going to slow down. I said it's going to slow down the rate. What is rate? Come on now. Slope, right? So if you were going to graph this, you'd put time over product, substrate, product. Oh, man. Okay, so let's say the red is normal. That's how fast amylase works normally? Wait. Okay, so that's normal. That's how fast amylase works normally. So if the pH is wrong, it's the blue line. pH, temperature, or salinity is wrong. It just doesn't work as good. So it slows down the rate if it's not right. Okay, right here on the red one, I went back and changed it. Look at the purple line now. Why did it top off? Yeah, it was either it's the highest it could work or what? You ran out of substrate. It's all gone. Like when you eat a cheeseburger, your amylase is breaking. Okay, eventually it runs out. Unless you eat another hamburger. The carbohydrates in your mouth are gone. Right? So it tops off. Y'all remember that? So you got non-competitive and competitive inhibitor? Yes? Okay, what I want you to do is work on this so you don't have quite as much homework. And talk about it. Ask me questions. This is your chance. So tomorrow, we'll finish the questions, do the multiple choice questions in class, and take the quiz.
Which question? I mean, finish these.